All right, first things first, let me start off with a little explanation and possibly an apology. I've been having technical difficulties, of course, with my iPad, and after a bad take on digestion 1 of 5, I thought it had been deleted. It may have been uploaded, and there might have been some inappropriate comments at the end, and for that I apologize. Also, the last ones that I had done were not uploaded. It had to do with a memory system or memory issue and storage issue. I just recorded a couple more that didn't record because of that. So with that being said, I want to try the next 3 videos and attempt to get through digestion to be able to get that to you today, which hopefully I will be able to do. Now that I've got that small disclaimer out of the way, let me go back to where I want to pick up. Hopefully those other videos are uploading right now, or they are on YouTube, so I can jump to this one. This is essentially where we were. And what we were talking about is really 2 important things. We were talking about 1, how do we actually break things down and digest things, and 2, how do we absorb them? We won't get to absorption on this video. We'll get to it down the line when we start dealing with the world of the intestines. We've had a little absorption here and a little there, little in the mouth, little in the stomach, but really the game changer is when we get to the intestines. I started out, and I'm going to continue on with this list of digestive enzymes. HCl isn't an enzyme, but it does put especially pepsin into its optimal pH for it to digest polypeptides or proteins down to polypeptides. Remember, the name of the game here is trying to break down the major biomolecules: proteins, carbohydrates, lipids or fats, DNA and RNA. Those are the things that we need to break down. We hit kind of the atomic, or not atomic, but the cellular structure. We looked at the parietal cells producing all of this HCl in these little tiny glands. So we've got a lot of surface area here. There's the mucus cells that produce that lining that helps to keep all of us from burning up from the inside out. We've got the rugae in here, which gives us quite a bit of surface area in here. So we've got mechanical as well as chemical digestion going on in the stomach. Then I think the last thing I put up, last slide, was this. We talked about HCl and how things like Nexium can block these pumps. But to go on from here, we're going to enter into the intestines, and the initial component of the small intestine is called the duodenum or the duodenum. Either one of those, both of those, is acceptable as far as the pronunciation of the first portion of the small intestine. But in order to take the contents from the stomach and dump it into the small intestine, we need to change the chemistry, and we need to change it dramatically. And that's where we get into this thing called a biliary tree. We've got 3 different organs that are often considered accessories. You can see the duodenum here, the duodenum. Stomach would be up in this area, which we don't have because we're focused on these 3. We're focused on the liver, we're focused on the gallbladder, and on the pancreas. Now, it's not just about putting contents into the intestines. It's not just about adding material. We're going to see that with the liver, we're also involved in cleaning the blood. With the pancreas, we're also involved with doing things like regulating sugar levels. So we've got a couple things to add with each of those. But I'd like to start out with the liver, this big organ that has 3 lobes on it that falls just below your diaphragm to kind of give you some orientation of where this is. And if you look, we've got all of these green lines that are intersecting and coming down in here into the duodenum. We have to be able to look at what's the cellular structure that's going to secrete contents into these, and what are these? What's the green stuff? You're going to see the green coating in the next few slides as well. The green is what is called bile, B-I-L-E. Bile is going to play a pretty important role in digestion of fats and lipids. And that's secreted in the liver. It's produced in the liver by some interesting cells called hepatocytes. You'll see hepatocytes in the next couple slides. They all start to come together into the left hepatic duct and the right hepatic duct. And then those are going to converge into the common hepatic duct. And the common hepatic duct can take a quick little right-hand turn into the cystic duct that goes to the gallbladder, the second thing that I want to talk about, which is just a storage bag. It's not that exciting. And then it will continue with the common bile duct. And then what you can't really see here, it's just this little stump. It's the pancreatic duct, which actually goes all the way up to the tail here. And it's going to bring in more contents along with the bile. So we have a number of things that are all going together, but what we want to focus on is the fact that they're all going to dump in and intermingle, co-mix with what's coming out of the stomach. Remember, low pH, and we're doing a lot of digestion there. So let's take a look at this in a little bit more detail. You can see upper left, we've got the liver with the 3 lobes. And these lobes are made up of what are called lobules that might be reminiscent of osteons if you think all the way back to compact bone in the skeletal system. Each one of these interlocked lobules, if I blow it up, we see right here. And in a perfect world, they're all hexagons. They're not. Some are pentagons, some are more squarish, like oblong diamonds, but typically we'll see them represented as hexagons. And a couple of things that might stand out. We've got this central area called the central vein. That's what we can see in the middle of each one of these hexagonal columns. And then we've got these kind of complex areas out around the outside. And these are called hepatic triads. And it's this. I'll show you hepatic triads in another slide or two. And then going in and going out, we see all of these spokes. It's a spoke-like network coming from the triads and going to the central vein, or conversely, going away from the central vein area to the hepatic triad. The business portion are these cells right in here. These are the hepatocytes. These cells are going to be cleaning the blood. They're going to be producing bile. They're really the heavy lifters in the liver when we get right down to this organ. So here's what's going to be happening with this. The portal vein is going to be coming in, and it's going to be sending blood through, and the hepatic artery is going to be commingling here and sending blood through. The two of them are going to be supplying oxygen, and they're going to be supplying nutrients to the hepatocytes. Now, when they do that, it's going to allow the hepatocytes to also take toxins and medicines and alcohol and other things and remove it, get rid of it as waste, break it down. But if you look, these arrows in the blue and the red are all going in towards the central vein. Ultimately, this is going to leave through the hepatic vein. But if you look at the green is moving away. It's moving towards the outside. The green is what now contains bile. That bile from these little tiny microscopic tubes in here will start to come together into the right and left hepatic duct and into ultimately the common. So directional, we've got 2 different directions going on in here. One, O2 and nutrients coming in. We've got the breakdown of toxins and the cleaning of the blood. And then in the opposite direction, we have bile production. This little computer-generated schematic is what, in an ideal world, this would look like. Dead end bile ducts or ductules coming out, and then we've got our vein and our artery going in and coming right into the central vein. All of this clear white area, it's all hepatocytes. Those are all the cells doing the job. Here's what it really looks like. Told you they weren't all hexagons. I mean, this lobule, we have 1, 2, 3, 4, 5. I guess it's a pentagon. But the important thing is that we can see a central vein here. These triads are out here. And hang on, I'll show you a triad in much higher magnification on the next slide. And then you can see all of these lines coming out and in, going in with blood and going back out with bile. It's not clear, it's not green and red and blue color coded on this because this is an actual slide. Let's zoom in on a triad. I learned them as a hepatic triad. This slide that I have procured is calling it a portal triad. Either one will work, just like duodenum, duodenum. And what we see are three things. Portal vein, or branch of, hepatic artery, these are bringing in your O2 and your nutrients, and flowing in towards that central vein. And then here we have a branch of the bile duct. So the bile is coming back out and dumping into the bile duct, ultimately to the right and the left, and then to the common, and then it will overflow into the gallbladder. So that's what we see essentially happening here. Clean the blood and produce bile for digestion. Now if we go back to this image, what we see is essentially all of this bile coming together, and it will overflow into the gallbladder. Now the bile, a big role of the bile is going to be to help in the breakdown, or what we call to emulsify. I'll show you that term in another slide or two, to try and emulsify fat, emulsify lipids. We've still got the pancreas coming down here, so we'll deal with that in the next, video, if it all works out for me. When we have fat in here, when you've just eaten a fatty meal, what the gallbladder will do is contract, and it will push this bile in. So the bile is going to be ready to help with the breakdown of that fat. So we've also been dealing with the fact that we've got to play around with the chemistry, right where I started this video out. So here's where I want to end this, with about a minute left, and hopefully I've done this correctly. I'm going to add on to this, and what bile does is it emulsifies fat. Now it's not a true digestion of fat, but you guys know surface area is important. If you want to attack something, you need a lot of surface area. So if I have use a very scientific term, a glob or a blob of fat, of lipid, if I can break it into a whole bunch of smaller droplets, and I could do this to be the same size as what's on the left side of the arrow, and then I was to ask what one, left or right hand side, has more surface area, I think you would all conclude, ooh, the right hand side has a lot more surface area. And I would say, you're correct. Which means the chemistry that's going to follow down here is going to be able to attack these lipids, that fatty meal that you just had, and be able to break it down more efficiently and more effectively. Bile also, in my last 1 minute, does a couple of other important things. It's going to increase the pH. Remember, coming out of the stomach, you're looking at a pH of 1.5 to 2. Not good when you get into the intestines. Also, lastly, bile will help to deodorize feces, which is always a good thing. So, emulsify fat, de-stink feces, and change the pH. And that's when we start to bring in the pancreas. So, hopefully this worked this time. And next time, on this next video, we will start out back with the biliary tree, and we can get into the pancreas.